Hi everyone, thank you for joining. My name is Tommy Varley and I'm a student at Case Western Reserve University. I'm a member of the YChromosome lab and today I'll be presenting our work on investigating the origin of cells at the maternal and fetal interface. During human pregnancy, the placenta is a vital organ that provides oxygen and nutrients to a developing fetus. The placenta and surrounding regions consist of cells of both maternal and fetal origin. The interactions between these cells are important in understanding human labor. Thus, identification of cell origin poses an important first step in understanding this relationship. Single-cell RNA-seq data provides us a rich opportunity to explore cell origin. The goal of this project was to explore the breakdown of maternal and fetal cells in this tissue and compare the breakdown across placental locations and cell types. We utilized 3 different methods to differentiate between maternal and fetal cells. In the first method, we examined Y chromosome-related gene expression in samples where the fetus was male. In the second method, we utilized Trumuxla, a tool to demultiplex pooled samples. And in the third, we trained and utilized our own model, placenta, to infer cell origin. We utilized the dataset from Trumuxla et al. This dataset contained 25 libraries across 9 pregnancies, with samples from differing clinical states and placental locations. Clinical states included preterm in labor, full term in labor, and full term not in labor. Placental locations included the placenta, the chorioamniotic membrane, and the basal plate. The read files were processed using the 10x Genomics Ranger package. Pre-processing of the gene matrices, the gene expression matrices, then included filtering by cell and gene count thresholds, as well as mitochondrial gene thresholds. We normalized and log transformed the data, as well as regressing out variation from total and mitochondrial counts. To cluster the dataset, we used the Leiden algorithm, and then used marker genes from Trumuxla et al. To assign cell types. The first method we utilized to identify the maternal-fetal breakdown was using the Y chromosome-related gene expression. We utilized male fetus samples where we would expect the maternal cells would not exhibit Y chromosome-related genes, and fetal cells would express those genes. The Y chromosome count ratio refers to the number of Y chromosome-related genes divided by the number of autosomal genes expressed for a given cell. We also applied this methodology to the female fetus samples, where we would expect that no cells would be examining any Y chromosome-related genes. In Figure 2, we can see that our expectations were confirmed. The second method we implemented was Trumuxla. This tool utilizes single nucleotide polymorphisms to demultiplex pooled samples. We utilized known cell types that we knew were either completely maternal or completely fetal to then identify the demultiplex samples as either maternal or fetal. The third method we utilized was our model placenta. Using the male fetus samples as training data and using the Y chromosome method to give labels to that data, we trained an XGBoost model where the features were the autosomal chromosome-related gene expression, and then the model then predicted whether a cell was maternal or fetal. The model achieved an AUC of 0.91 and an accuracy of 0.87. On the same set, Trumuxla scored an accuracy of 0.77. So note at this point, Trumuxla is able to be applied to all samples regardless of the sex of the fetus, whereas the Y chromosome method is only applied to the samples where the fetus is male. And placenta is only applied to the samples where the fetus is female, as it was trained on the samples where the fetus was male, and it largely follows what the Y chromosome method, those results. So next, we'd like to compare the performance of the tools across these different subsamples, and we do that by comparing that to known cell type expectations. So as you can see here, using a heat map where 1 indicates that the sample was 100% fetal cells, 0 indicates that the cells were completely maternal cells. And then you can see we have the different methods and the different across the different subsamples where those methods are applicable. And we have at the bottom here the expected predictions from prior knowledge of what cell types, what the breakdown looks like. So first, for cell types that are completely fetal, those include trophoblasts, which are the first 4 cell types in each heat map. And you can see that all 3 methods performed relatively well. One unreliable part of placenta was that for the STB cell types, there was only 12 cells of that type in the training data. So those predictions were largely unreliable coming from placenta from the lack of training data. Next, for cell types that are largely maternal, such as endometrial and decidual cells, Both placenta and the Y chromosome ratio performed very well in those cell types. Trumuxla performed well, but it did have some areas where, of poor performance, such as in endometrial cells, where it did predict some fetal presence. In cell types where there is a mix of both maternal and fetal cells, it's difficult to examine and determine the truth, as the truth is unknown that there's both types of cells belong in those cell types. However, we can compare and see that Trumuxla does predict more of a fetal presence in the cell types where both maternal and fetal cells are present, whereas the Y chromosome ratio and placenta show that there's a greater maternal presence. In conclusion, we've shown that placenta performed strongly with an AUC of 0.91 and an accuracy of 0.87, which outperformed Trumuxla, which had an accuracy of 0.77. Placenta matches the expectations for the known maternal fetal breakdown of cell types, as shown by those heat maps. One potential source of error in placenta is caused by using the Y chromosome ratio to create labeled data. Maternal cells are unlikely to express Y chromosome-related genes, but it is plausible that fetal cells did not express Y chromosome-related genes, thus introducing false negatives in our training data. An area we'd like to explore is the future importance of genes in determining cell origin. Many cell types are either all maternal or all fetal, so we'd want to explore whether our placenta is really picking up on the differences between maternal and fetal cells, or whether it's relying on finding the cell type and using those differentially expressed genes. Thank you very much for listening. I'd be happy to answer questions.